Hello, my dear students. This is your host, Dr. Syed Parker. Today, let us study about spectrophotometry and radiobiological techniques. What is spectroscopic technique? It is one of the most fundamental technique used in analytical chemistry to measure the concentration of a particular substance or solute in the solution. Every laboratory is equipped with this instrument because it is one of the most fundamental instrument required in different scientific horizons. What is the principle? Let us see. Here you have a light source and collimator. This is nothing but a lens. This lens captures or assign the light towards monochromator prism, and from this different electromagnetic radiations are emitted. And here you have wavelength selector. This wavelength selector a particular range of electromagnetic radiation, for example UV visible radiation or visible range. This range is selected and passed through the sample, and this sample absorbs the specific wavelength or absorbs the light, and it again emits the light which is captured with the help of photocell or detector. This is further given in the form of digital reading or digital display. This entire uh, construction is based on Beer-Lambert's law. So what is this Beer-Lambert's law? Here you can see I denotes to the amount of light, the amount of number of photons emitted per second, which is nothing but the amount of light per second. And here you can see I0 and I. I0 is the intensity of light before it could travel through the sample, whereas I denotes to the intensity of light after the sample. So this is extinction coefficient. D is nothing but the thickness of the cuvette, which is usually 1 centimeter, and concentration. So the absorption is equal to log of I divided by I0, which is nothing but the extinction coefficient into thickness and concentration. So this complete Beer-Lambert's law give rise to the mechanism and principle of spectrophotometer. Here you can see the mechanism of a spectrophotometer with light source, which we already discussed, monochromator, sample holder to hold the cuvette, detector to measure the intensity of light transmitted, signal processor which gives the result in the form of signal, a readable format. Moving on to the application properties of spectrophotometry, it is one of the best instrument for quantitative analysis in order to measure the concentration of a substance in a given sample. And they are widely used in the molecular biology and microbiology in order to determine the growth pattern of bacteria, in order to see lag, log, stationary and decline phase, whereas in molecular biology it is used to detect the concentration of nucleic acid, proteins and other compounds. Biochemical analysis, they are used in enzyme assays, protein concentration measurement, etc. Further, in pharmaceutical industry, they are used to determine the drug concentration in pharmaceutical formulation. Environmental analysis, it is also used in monitoring the environment quality of water, etc. And in industry, in order to maintain the quality of desired products, especially in food and beverages, it is used to know the concentration of specific compound. So like this, spectrophotometry becomes the most fundamental instrument, and there are different types of spectrophotometry which can be seen, which as UV visible spectrophotometry, IR spectrophotometry, etc., which is divided based on the electromagnetic radiation involved. So moving on to the second part of this lecture series is radiobiological techniques. Radiobiological technique involves the study of the effect of ionizing radiation on living entity or living organism, with the focus of understanding how this radiation can create impact on living organism, whether it can damage or whether it can help in developing strategies to mitigate or exploit this radiation for beneficial aspects. This radiobiological techniques are essential tool, especially in the field of medicine, radiation therapy, especially for cancer patients, environmental science, nuclear research, biophysics, etc. So, what are the characteristics of radiobiological techniques? As I said, majority of researchers are practicing the radiobiological techniques in order to determine and investigate the biological response towards ionizing radiation. Sometimes they use a particular cell or particular tissue in order to study. In some cases, they take the complete animal. The whole animal is exposed to radiation for a specific period of time, and that after the specific period of incubation with the radiation, its systematic effect on living organism and its behavior is studied. Under radiobiological techniques, you also have dosimetry. Dosimetry is nothing but the measurement and calculation of a radiation dose to ensure accurate and controlled exposure in a particular experiment. This is usually practiced in medical application. Biological dosimetry, under dosimetry, is nothing but assessing the biological effect of radiation, often through techniques like chromosomal abbreviation analysis, which helps the researchers to understand the impact of radiation on a living tissue. Further, radioprotectors and radiosensitizers. Research also focuses on the substance that can protect a normal tissue from getting damaged from the radiation, the exposure of the radiation. Such substances are called as radioprotectors, whereas there are certain compounds which enhances the sensitivity of target tissue, and these compounds or substances are called as radiosensitizers. Further, moving on to the important character of radioisotopes. These are the isotopes made of chemical elements that exhibit radioactivity. Some of the characteristics of radioisotopes include radioactive decay. Radioisotopes undergo a spontaneous radioactive decay by emitting radiation in the form of alpha, beta, and gamma rays to achieve a stable state. This decay is steady based on the applicative property. So half-life, this decay influences the half-life of this radioisotopes. Each radioisotope has a characteristic characteristic half-life, which is the time required for half of the sample to undergo radioactive decay. This characteristics is crucial for various applications including medical imaging of sample and also analyzing geological sample, archaeological sample, etc. Specific decay pathway. Each isotopes undergo through a specific pathway, and this pathway depends or influenced by the type and energy of radiation emitted. When it comes to application of radioisotopes, they find their application in various fields such as medicine, especially diagnostic imaging and cancer treatment. All these radioisotopes or radiobiological techniques are majorly used in cancer therapies. They are also used in the determinant of contaminants in industry, and they are also used for sterilization of particular object or particular space, etc. Radio autoradiography. Autoradiography is a technique that uses the radioactive emission from a labeled substance, usually radioisotopes. And this radioactive emission creates an image of distribution of the substance within the biological sample. The principle of this autoradiography is that a sample, for example, tissue or section of a tissue, is exposed to a radiation emitting substance, and this emitted radiation exposes a photographic film or photosensitized detector. Once the radioisotope is exposed to a particular tissue or a section of a tissue, it emits the radiation in the form of photographic film. So this complete process is called as autoradiography. Radioisotope dilution technique. It is one of the important technique practiced in various cell biology and chemical biology and cancer biology. Here, the radioisotope dilution technique, it measures the quantity of a substance in a sample by introducing a known quantity of radioactively labeled form of that substance, and then measure the dilution of the label as it mixed with unlabeled form. First, they introduce a known quantity of radioactive labeled substance, followed by its measurement with undiluted, unlabeled mixture of radioisotopes. So this mixing between labeled and unlabeled form gives information about its decay or about its other applicative properties, especially in chemistry, biology, and medicine. Pulse chase experiment. It is the experiment which is commonly practiced or applied in cell biology and biochemistry in order to study the kinetics and fate of biological compounds or biomolecules within a particular cell or particular tissue. For example, proteins and nucleic acids, how they perform, what is the shelf life of it, how are they influenced with the exposure to various other environmental stress, etc. Here, this experiment involved two steps. The first main step is pulse phase. In this phase, labeled precursor is introduced into the cell, followed by chase phase, where cells are provided with excess of unlabeled precursor. In first phase, a labeled precursor is introduced, followed by providing excess of unlabeled precursor. This technique helps researchers to understand the synthesis, movement, and degradation of specific molecules within the biological system. So moving on to the basic principle of absorption and radiation. As we discussed, the absorption of radiation, especially the spectroscopic technique, it involves Beer-Lambert's law. And here the basic principle is use of electromagnetic radiation, and atoms or molecules within this medium absorb energy from incident electromagnetic waves, or creates analytic tool or becomes the analytic tool in order to measure the concentration of a solute in a solvent. This is also used in determining quantum mechanics. Moving on to the applicative property, the first application is in diagnostic imaging, medical imaging. In recent times, it is also used in order to predict the climatic changes by measuring the greenhouse gases absorbed and emission using infrared radiation, which is very important in order to maintain the earth temperature. So with this, we have completed the overview of radioisotopes, radiographic techniques, characteristics of radiographic techniques, and we have also overviewed with the spectroscopic technique, the principle of spectroscopy, the applicative property, how it works. So with this, let me conclude this class today, and I wish you all the best. If you have any doubt, you can contact me anytime. Good luck.